thank you so much for accepting this invitation. This is Amit Sharma from National Institute of Malaria Research. I'm introducing myself because we have participants, literally attendees from all across India, from structural biology backgrounds or otherwise. It's a really special privilege for me to host Professor David Stewart for today's distinguished lecture talk. I think all of you have seen the flyer in a series of papers this year, the breakthroughs that have been made for in our understanding of COVID-19, and I can assure the audience that the work you will see will be beyond cutting edge today, and it's really going to be very, very illuminating and inspirational for all of us. I will very briefly read Professor Stewart's CV very briefly. Many of you already know about him, but just to highlight a few very critical aspects. So Professor Stewart is a MRC Professor of Structural Biology at Oxford and also head of the Division of Structural Biology in Clinical Medicine in Oxford. He is also currently the Director of Life Sciences at the Diamond Synchron Light Source, which you know is one of the best light sources in the world. And he has been the director of that since 2008. We would like to congratulate Professor Stewart because this year he was awarded a knighthood. He was in the New Year's honors list in acknowledgement of his work as one of the key responders to the COVID crisis and his pivotal role in understanding of COVID-19. But as you will see, today's talk is focused on COVID, but of course, Professor Stewart has been working with virus crystallography and virus structural biology for many decades. He's a well-known international leader in structural biology. It all started off in 1989 with the structure which was on the cover of Nature, the foot and mouth disease virus structure. And then there were a series of breakthroughs, structures of the blue tongue virus cores and the PRD1. These are some of the largest structures solved today, and also of the first envelope virus protein structure that was solved in his lab. And if you go through his publications, which are over 375 publications, he has really made tremendous contributions to our understanding of polio virus, HIV, hepatitis A, B, SARS, and Ebola virus. And the most recent award that he received was the Royal Society in Gabor Medal in August 2020. And this was for his seminal contributions to our understanding of the foot and mouth disease virus, the polio virus, and COVID, as in the SARS-2 virus. So it's a really great honor for all of us to host you here, Sir Professor David Stewart, and we look forward to our talk. Thank you.
So thank you very much for the kind introduction, Amit, and it's a real honor to be asked to give this talk to everyone. I have no idea who's in the audience, but as Amit said, I'm a structural biologist, and what I'll be talking about is a little bit sort of broader than pure structural biology today. So the title of the talk is The Changing Antigenic Anatomy of SARS-CoV-2. And let me see. Yep, that's good. So 18 months ago, the virus was, well, it's not 18 months now, it's about 20 months ago. The virus was essentially unknown. And it had been a long time since I'd worked on any coronaviruses. And now loads of people around the world have very strong opinions on this virus, and it's probably the most intensively studied virus of all time. It seems to be overtaking HIV at the moment. And I've been personally lucky because being in the Department of Clinical Medicine in Oxford, where there's been a lot of really good work being going on, including obviously the vaccine work of Sarah Gilbert and Andy Pollard and groups, and some excellent immunologists. I've been able to collaborate with people on trying to understand, in particular, the antigenic side of this virus. So I'll see how I get on with the talk. I put together a talk that's different to I've given before, so I may misjudge the time. I'll give a couple of slides of introduction to the pandemic, introduction to the virus, talk about the antigenic anatomy of the early pandemic virus, and then talk about the impact of variants on antigenic responses and briefly mention the changes that occur in the antigenic responses in response to these variants. And then if there's time, I'll just sort of try and say something about where we go next, or where the virus might go next, and how we might respond. So I just wanted to make the point that there's been a tremendous response from basic science to the pandemic, and it's been in many areas. Certainly in my judgment, the work that we've been involved with, the molecular side has been absolutely outstanding. Where things have got slow and difficult has been in stitching together perhaps some of the molecular side with other activity, including animals and clinical trials. So I think there is an argument that we should be really thinking hard about how we set up, whether it's nationally or ideally in a sort of global way, thinking about pandemic response pathways. And in response to a call from the European Union for grants in this area, this is an application that has now, in the last week or two, just been awarded by the EU. With others, we were involved in trying to map out what is necessary to respond to existing and future pandemics. And I just wanted to, a lot of this is obvious. It involves social sciences, it involves epidemiology, it involves basic virology, immunology, structural biology. But I just wanted to make the point, I won't labor it by describing the impact, but structural biology has actually played a role in the present pandemic at a whole variety of stages, from characterizing the host factors, the virus factors, the characterizing the pathogen, helping develop assays, drug development, vaccine development, and diagnostic development, and I hope to some extent in understanding the emergence of variants going forwards. The thing to bear in mind is that the results came very quickly, but they were built on many years of prior work. So there was existing knowledge of the proteins of SARS and MERS that was very helpful in kickstarting things very quickly. So underpinning basic science is really important if we're thinking about how we move forward in this. In terms of where we are with structural biology, yesterday I did a search of the protein data bank, which is where all of the public protein structures are deposited, and at that point there were approximately 1,400 structures, and of those, the large majority, about 2/3, have come from crystallography. So the big high impact methods at the moment in terms of new structure determinations in structural biology are crystallography and electron microscopy. Electron microscopy is very powerful, but we've got to remember that crystallography has a tremendous history to it and is very effective. And in fact, the majority of what I'll talk about comes from crystallographic investigation rather than cryo-EM. So both methods have a strong role. So what I'll be talking about is really a single protein. So the SARS-CoV-2 genome is large. So these coronaviruses are some of the largest RNA viruses with about 30 kilobase genome. The spike protein, S, is a large protein of over 1,200 amino acids. It forms a homotrimer, which is embedded in the virus membrane and has more than one function. And this protein is the target of vaccines. The majority of vaccines are really focused, in many cases exclusively, on eliciting antibodies against the spike. And indeed, if we look at the distribution neutralizing antibodies, they are focused very heavily on the spike and on particular parts of the spike. The spike is large, but there are different portions of it, and one important portion is called the receptor binding domain, and there are 3 of these because it's a trimer. I've just shown one here. This is right up at the top in the middle of the spike, and there's also an N-terminal domain, which is also quite important, although we know very little about what the function of that is. And I think it's fair to say that the N-terminal domain is undergoing very rapid change and evolution at the moment as this virus adapts to its new host, which is us. So this is just to give you a picture of one aspect of structural biology contribution. So this is cellular tomography applied to cells that have been infected with the ChAdOx1 vaccine, which very large amounts of this have been produced in India, of course, and in other places. So here the adenovirus has infected these cells, and just a few hours after infection, and this work is led by Pei-Jun Zhang and Sarah Gilbert in Oxford. Recently, our group have also played a role in some of this. But the key thing to realize here is that the infection of the adenovirus leads to the production of very large numbers of spike proteins, which are produced on the surface of the infected cells, and it's these that elicit the antibody responses. And what's nice about this analysis, I can't see my mouse, I'm afraid, but so I can't point them out, but on the picture on the left and the right, you can see the cell membrane, and beyond that are the trimeric spikes. And it's clear from this analysis that these spikes are in the pre-fusion state. So the spike is essentially a fusion machine. Its job is to, first of all, recognize the cell receptor, and that recognition is done through the receptor binding domain. The best defined and essential cellular receptor is ACE2, and so that binds to the receptor binding domain at the top of the molecule. Prior to fusion, there's a cleavage of the polypeptide chain of spike into 2 subunits, S1 and S2. S1, where the receptor binds falls off at that point, and S2, which forms the stalk of the molecule, does a massive conformational switch to engage a fusion peptide in the host cell membrane and cause fusion and infection of the cell. So that's the sort of story of the protein. It's a large protein. It's also very flexible. And in order to study it, there have been mutations introduced to increase the stability, or in some cases to decrease the stability of the post-fusion state, so that the protein remains in the pre-fusion state. The pre-fusion state obviously is the only state that can bind the receptor, and that state is important for generating neutralizing antibody responses. The flexibility is in part movements of the receptor binding domain. On the left there, I've indicated the receptor binding domain in the up conformation in one case, and in 2 other cases, the receptor binding domain down. So this is a major rotation, a swinging of this domain up and down. My thought is that when the virus is released from cells, the receptor binding domain is in the down conformation, and it's stabilized there by lipids that are incorporated from the host membrane as the protein leaves. The loss of those host lipids then increases the flexibility of the molecule, increasing the presentation of the receptor binding domain in the up conformation. So only in the up conformation can it recognize the ACE2 receptor, and the complex of the ACE2 receptor is shown on the right there. That's based on a crystal structure, and The footprint of the ACE2 on the receptor binding domain is very small. It's a couple of dozen residues, but a huge amount of biological function is focused in this region of the spike. So a massive molecule with a tremendously important collection of a couple of residues, a couple of dozen residues on the tip of the receptor binding domain. So what I will talk about in the remainder of the talk is a single, very close collaboration with a group that works adjacent to us in the Wellcome Center for Human Genetics. And that group is the group of Gavin Screeton and of Dew. So what we've been working with them on is to dissect the antibody responses. The first thing we did was to isolate antibodies from 42 recovered patients, mainly in the Oxford area. From that, Gavin's lab were able to isolate 377 anti-spike antibodies. From those, 80 antibodies have been looked at in considerable detail. Those are the ones that bind the receptor binding domain, and one further antibody we've looked at in detail. But I won't go through the methods of obtaining the antibodies. It was pretty standard cloning from single B cells. A couple of different methods were used to give that set. So if we look at that set of 377 antibodies, they really show that there is a polyclonal B cell response against this virus in these patients, in these convalescent patients. One of the things that was immediately obvious was that the number of somatic mutations in general was rather low. So you can see at the bottom there that the heavy chain and the light chain both had approximately on average, four mutations, somatic mutations away from the original germline sequence. So this reflects the fact that these antibodies didn't come from a pre-existing repertoire in response to another coronavirus infection. They were generated from germline and antibodies pretty rapidly here. The majority of these antibodies really had nothing to do with binding the receptor binding domain. They were spread across the molecule. Only about a fifth bound the receptor binding domain. However, when Gavin's group looked at the potency of these antibodies, the non-receptor binding domain antibodies were generally very weak neutralizers, with a single exception, antibody 159. Whereas the receptor binding domain antibodies, a good number of those, and you can see at the bottom, over a quarter of those were really potent neutralizing. By very potent, I mean less than 0.1 micrograms per mil IC50. And the importance of the receptor binding domain binding antibodies in the polyclonal response is confirmed by this experiment, where from the complete polyclonal serum, with a full set of responses, essentially the receptor binding domain was used as bait to pull out antibodies that bound strongly to the receptor binding domain. This was just the receptor binding domain on its own, so some antibodies that may be missed because it may not be perfectly presented, but it's a very good approximation. And you'll see that between up to 90% and down to about 55%, depending on the patient, of the neutralization came from those RBD binders that were absorbed out when you went in with bait of the receptor binding domain. So the large majority of the neutralizing antibody responses are against the receptor binding domain. When you look at the sort of genetics of the response, what we and others, particularly Ian Wilson's group and Bjorkman, various other people have found, and this is generally true against responses for responses against the early pandemic virus. There are some variable gene families that give very strong, high potency neutralizing antibodies. And the blue arrow marks the 353 gene family, which where nearly all of the antibodies and a significant proportion of the total number of potent neutralizers belong to this gene family. So this is then a public response. Most people will make antibodies that belong to this gene family that are potent neutralizers. This public usage of neutralizing antibodies has been seen in other diseases. So we've been working now for ages, and we've done dozens of structures by cryo-EM and by crystallography, as I say. And I should say that as well as our groups, there's been very strong support from the Diamond staff at the beamlines and also on the electron microscopes at Diamond. Diamond, in the early days of the pandemic, very early on, set up emergency response to the pandemic so that people working on SARS-CoV-2 related work could get accelerated access, and that's worked very well. So various things came out of all of these structures. Even before we got the structures, one thing was clear, and that was that the large majority of the potent neutralizers block ACE2 binding. And that's true with ELISA assays and also with biolayer interferometry biophysical assays. So I'll just mention a couple of things that came out of this, because you get, one of the nice things about structural biology, you get this sort of rich set of information, and whether you can make sense of it all or not is another question, but it does throw up sort of interesting things. So I'll just mention one thing, which this is something that is well known, but almost 20%, well, it's well known in the field of antibodies, but little discussed, I feel. Almost 20% of the 80 receptor binding domain antibodies that we looked at are glycosylated. We all know that antibodies are glycosylated in the constant region. That's structural. It helps the antibody be stable. But these were glycosylated in the complementarity determining regions. And we've found that the glycosylation can occur in the light CDRs, light chain CDRs, and the heavy chain. We looked at crystal structures of 3 of these. One of them was in the CDR1, one of them is in CDR2, the other in CDR3 of the heavy chain. In none of these cases does the sugar make a massive difference to the binding affinity, but in several cases, if you remove the sugar by mutation of the sequence, then you reduce the effectiveness of the antibody to some extent. And in all cases, bizarrely, and we don't know the reason for this, the sugars wrap around one particular region of the receptor binding domain, and we call that the left shoulder. We'll come back to that. So we don't know the reason, but it's interesting. This may be telling us something about whether there may be interactions of the receptor binding domain naturally with sugars. There may be some other biological function going on here that we don't understand. Another thing we looked at, which I was surprised at the outcome, was swapping the light chains. I've mentioned that there were several of these public responses, and that means that there was the heavy chain had the same variable region, but the heavy chain was paired with different light chains in these public responses. So Gavin's group made antibodies where the heavy chains were kept fixed, and the different light chains from the different representatives of the public responses were matched with each of the different heavy chains. And what was interesting was that in one case, it was possible to identify a favored light chain, which, when that light chain was combined with the slightly different heavy chain versions of the different antibodies in that gene family, it essentially produced a 100-fold improvement in the strength of binding. And that was, when we looked at the structure, it was quite obvious. The mutation gave a larger hydrophobic residue that stabilized attachment to this part. And again, this is to this left shoulder of the receptor binding domain. So, okay, let's now just stand back and do a survey of this. So we've got 80 antibodies that bind to the receptor binding domain. We've got 20 potent antibodies that bind to the receptor binding domain, one that binds elsewhere. That's 80 is too big for us to contemplate doing structures for all of them. So we thought of a different way of doing it. But that was focused on the receptor binding domain. Before saying that, I'd just like to sort of mention the N-terminal domain. Occasionally, there are potent neutralizers that bind the N-terminal domain. And from the picture here, you can see that that is on the outside of the trimer. Here we see the one non-receptor binding domain antibody from our set that was potent, and that binds this N-terminal domain. We've got no idea of what the mechanism of neutralization of this is. It's really that remains a mystery. The binding sites are too far away for any direct sort of steric effect. They're too far away for bivalent attachment. We just don't know. I'll come back a little bit to this, but to be honest, the N-terminal binders are still a mystery. But it's obvious it's a different mechanism, because actually this antibody it neutralizes in a live virus assay, but it does not neutralize in a pseudovirus assay. It suggests that there may be something different about the way the spikes are presented, and that presentation of the spike might be important for the mechanism of neutralization, but that's guesswork. And it's important to say now, because I'll gloss over it later, that there are some receptor binding domain antibodies that don't block ACE2 binding, and there are well publicized examples in the literature. One is it's called CR3022, another is EY6A. There are others. So there are some antibodies that bind, that don't compete with ACE2, but still manage to neutralize. But often, though, that neutralization mechanism is a bit unreliable to detect with many neutralization assays. So there's been quite a lot of argument about this. But I think now it's well established that some of these antibodies can be effective in protecting against disease. So we have this problem then. We've got this 80 RBD binders. How do we make sense of them? And what we wanted to do was to map where they all bind onto the receptor binding domain. And here, this is where the left shoulder comes from. You can imagine that the receptor binding domain is roughly in the shape of a classical torso, and that can then allows you to define left shoulders. And the left shoulder is on the right in this view, but it's the left shoulder of the person whose torso it would be. And chest, right flank, left flank, and so on. So that gives us a way to navigate where the binding is. At that point, as I say, we wanted something that was higher throughput than just doing the structures of them all. So we brought on a biophysical method, which is biolayer interferometry. And what we do is you have a little tip, which you can chemically attach your RBD to the tip in the machine, and you measure by interferometry the reflected light reflected off this surface in the tip. When there's something bound, you get a signal. So you can see on the bottom, there's a signal when you bind RBD. If we go in with an antibody, we get a further increase in signal. If we now go in with a second antibody, we can ask the question, can that antibody bind at the same time as the first one? Because if it can bind, the signal will increase. And by looking at the ratio of these last two signals, we can get a numeric value for the competition between these two antibodies. Now, in principle, this can be done at very high throughput. There are 384 well versions of the machine. We didn't have one. We had a 96 well, but we had Daming, a postdoc in the lab who worked extremely hard to do this work on this machine. And essentially what he did was to do the competition between nearly all pairs of these 80 antibodies. And this shows the results in the random order that the antibodies were isolated in. The red dots mean that the antibodies compete, blue means they don't compete, grey means we didn't do the measurement. So at this point, Helen Ginn, who is by training a crystallographer and had recently developed a cluster analysis method based on single value decomposition, looked at this data and used cluster analysis to rearrange the matrix. And at this point, you can see on the picture on the left is the representation of the single value decomposition results from the cluster analysis, colored according to separate groups. And those groups are not, they do overlap to some extent, and you can see that in the competition matrix on the right. The blocks are not completely independent of each other, particularly the top left-hand blocks have some cross competition between them, but there are clearly discrete areas which we would call epitopes, and the coloring there on the bar down the side matches the stuff on the left to the right. So that's fine. We can bin the antibodies into different epitopes. Lots of people have done that, but what I wondered was, can we take this further and actually get structural information from this competition data? So Helen at this point essentially made a mesh, shrink-wrapped a mesh onto the surface of the receptor binding domain. Oh, there's a little window come up, which I can start annotating. This is too complicated, right? At least my mouse is working a bit now. So we don't know where the antibodies bind, but we know they will bind on the surface, which is represented as a mesh. However, we had some structures. So in our competition matrix, we could add in some structures where we knew where they bound and then anchor those on the surface, and then use a very simple sort of Monte Carlo and steepest descent method to move the other antibodies around to match the measured biophysical data. And oh heck, I'll close that, and then I can more easily move on. So we spiked with the data. We scattered all of the unknown antibodies around, and as I say, we did a Monte Carlo, which allows us to see whether it works reliably. Also, we had some known structures that we didn't put in. We kept those as secret from the method, and that showed that we could get about a 7 angstrom accuracy in the positioning. And now we can actually physically assign these epitopes to regions on the receptor binding domain. We can call them left shoulder, neck, right shoulder, right and left flank. Furthermore, then if we color by this epitope, we can position those on the receptor binding domain, and we can see that indeed there are these epitope clusters, and essentially these top 3 clusters in particular are very close to the green region, which is where the ACE2 binds. So now what we can do is to look at the neutralization power of these antibodies and color this picture by the neutralization ability. So blue means that they're rubbish neutralizers, red means that they're excellent neutralizers. And this really very starkly shows that nearly all of the potent neutralizers bind in a place where they will compete with the ACE2. It explains what we expected from the competition results. Of course, there are some that bind up in that region that are not very good neutralizers, and they're probably just weak binders. So the ACE2 probably outcompetes the antibody. So that gave us quite a nice overview of the antibody responses against the early pandemic, the Wuhan, the Victoria strain, whatever you want to call it. But then in late 2020, new variants started to emerge. They're now called alpha, beta, gamma, and delta. Some of the slides may still have the old naming schemes on, which those naming schemes were very hard to sort of follow. So I'll try and use alpha, beta, gamma, delta. As you know, the alpha appeared quite early on. It was first detected in September in England, in Kent. It was ignored not for quite a while, and then really came in with a vengeance. Beta has been a huge problem in South Africa, gamma in Manaus region of Brazil, and delta has been devastating for you in India, and is now a major problem globally, and especially in the UK where it's taken over. So all of these have a number of changes. They've got of the order of 10 mutations within the spike. All of them have at least one change in the receptor binding domain, and they all have changes in the internal domain. And at least for alpha and delta, it's very well established now that they are markedly more transmissible. So delta, maybe 1,000-fold more transmissible and has outcompeted alpha. So what determines transmissibility? I should say, I don't think we fully understand. But what we're also interested in is what the antigenic properties of these are. And this, let's see if I can play this. This is just shows a time course with a 7-day moving window of mutations from the original virus as recorded in the UK sequencing effort in COG UK. And you can see in September, there was no reason to be worried about the 501, but there it does start to appear. And at that point, in October or so, it would be reasonable to pick it up with the benefit of hindsight as a problem. And at this point, after some time of just bumping along at a low level, it starts to pick up. And you can see by the end of the year, it's shooting up to become early in 2021 completely dominant. So one of the things that would be nice is, can we use structure as a way of helping us flag up, put a red flag up when we see these variants starting to emerge in the sequencing effort where it's going on. And if there's time, I'll come back to that, but there probably won't be time. So this just shows in South Africa with the beta variant, in England with the alpha variant, the rapid move to dominance of the variant virus over time. So this shows where the variant mutations in the alpha, beta, gamma, and delta occur in the receptor binding domain. What's notable is that they are all either within or adjacent to the ACE2 binding region. Now, I would say at this stage, we've got no reason to think that these mutations have arisen in response to their antigenic escape strategy by the virus. They may have arisen for other reasons, and one reason might be to increase the transmissibility, perhaps by improving receptor binding. So that there's various things that we haven't yet teased apart here. Like other viruses, as I said, there's mutations in the N-terminal domain. The N-terminal domain is said we don't really understand it. If you compare SARS-2 with SARS-1, the original SARS virus, the N-terminal domain in SARS-CoV-2 has a series of insertions in these regions, and many of the variant viruses have deletions in the same region. So that's why I said I think there's a lot of evolution going on, and I think there's a lot of work to be done if the virus has got opportunities for markedly changing the N-terminal domain to improve whatever it is that it affects. And this this is just to show the close interactions of ACE2, particularly here with the 501 mutation. And if you look at the surface where ACE2 interacts, this is the wild type in alpha, beta, and gamma variants. This residue is mutated to a tyrosine, and then makes very good stacking interactions with the ACE2. And when we did BLI affinity measurements, you can see that the off rate following mutation to tyrosine for ACE2 is reduced. So the ACE2 has a longer residency time on the receptor binding domain with the tyrosine mutation. So the N501Y is in the right shoulder there, and this shows the effect that the mutation has on the affinity of antibodies. If we, sorry, this one shows the effect of the mutation at 501, the alpha mutation, on the affinity of antibodies. And you can see that for a number of antibodies, the affinity is reduced, some of them really markedly. The red ones are reduced by 1,000-fold. So it's not a large number, but it's some. And what's interesting is that this dominant public response of the VH353 antibodies is really crippled by this mutation at 501. And the reason why it's affected is that one of the light chain CDRs, the light chain CDR1, interacts with residue 501. So this is really unlucky. Was it unlucky, or was it was that one of the drivers for the mutation? We don't know. However, we'll come back to this. This public antibody class can still recover from this mutation in a few rare cases. If we go on to beta now, the South Africa variant, this has 3 amino acid changes in the receptor binding domain, and these include a particularly serious mutation, E484K, which has a big change in electrostatic properties and makes interactions with a good number of our the 80 antibodies that we characterized. The third mutation, 417N, again has a change in electrostatic properties, but we don't think this would introduce a big effect on the recognition of most antibodies. There's a lot of discussion about this, and I haven't got time to go into the data, but it appears that actually 417 makes the virus easier to neutralize. Now, I think that's an emerging consensus now. So it's not an antigenic effect, it's an effect on the sort of fitness of the virus, completely separate. And it may be because a part of this may be due to it reducing the affinity for ACE2. But if you take the beta receptor binding domain overall, because of the 484 and the 501 change, it does seem that this binds more tightly to the ACE2. The off rate here is about an hour and a half. So it does seem that the ACE2 interaction is one of the sort of drivers here. And the beta variant has a much more serious effect on very many antibodies. So you can see on these graphs from the Wuhan through to the beta variant, there's a dramatic loss of neutralization ability. And now the same plot of effect on neutralization on these antibodies show that a lot of these are pink or red, indicating a pretty disastrous effect. The N-terminal domain, there's a lot of changes, and all I want to say is that those changes in general have an effect on those antibodies that neutralize through binding to the N-terminal domain. We don't understand them very well, but we do see that other people's antibodies and our 159 antibody, the binding is abrogated to most of these variants. So these are very sensitive to the antigenic variation. Now, as I say, the 353s generally are useless against 501, but we looked very carefully and we found that there was one of these that had a particular light chain that was resilient to binding at 501. And in fact, if that light chain is then put onto the heavy chain of all of the others, it restores the ability to neutralize the virus. So this is useless for pre-existing responses, but it does indicate that there may be some 353 response to mutant viruses that harbor 501. And I haven't got time to talk about it, but we've got very recent data for the beta variant responses that show that there's a small level of 353 very potent cross-reactive antibodies that are elicited against the beta variant. So to go on to the Delta variant, this is really radically different in terms of the mutations in the receptor binding domain. You can see the mutations here. There is a mutation at residue 452 and a mutation at residue 478. This is data from the COG UK, so this shows that what happened. It's now a little bit out of date. This has now dropped away to virtually nothing. Black is the alpha variant, and here you can see that Delta completely outcompeted it very rapidly and took over in the UK. So good news is that nearly all of the potential therapeutic antibodies from Vir, Regeneron, AstraZeneca, Adagio, and Lilly remain effective against the Delta variant. So therapeutic antibodies generally look good. And indeed, this is now showing the effect of the Delta mutations on the binding of our panel of representative pre-pandemic type virus antibody, early pandemic antibodies. Again, there are some antibodies that are seriously affected, but many of them are not so badly affected. Now, this is a sort of important message and really sort of brings together a lot of stuff from a number of papers. It's massively complicated, but what we've got here is what we're doing here. Sorry, my mouse is very sensitive, so it tends to jump around, make the slides jump around. What we've got are essentially the neutralization titer of the different against different viruses for different sera. So these sera are people that were infected with early pandemic virus, and we can see these people are they're able to best neutralize the early pandemic virus. The worst is the beta variant, which is more than a factor of 10 worse, factor of 13 worse. So there's a big damaging effect with the beta variant, and a relatively modest twofold effect of Delta. Alpha, this is people that have been infected with the alpha virus, and here you can see the alpha virus looks like it's actually quite a good one. It appears to be close to a sort of universal vaccine, if you like, because it gives pretty reasonable responses against all of these. Obviously, much worse, still much worse against beta, but it's the best balanced response that we have. But what's really alarming here is that if you look at people that have been infected with beta or with gamma, these are the responses. Sorry, that red circle there should be over 307. My fault. This is the homologous sort of challenge, and the beta antibodies are good against beta virus, but the beta antibodies are useless against delta, and the gamma antibodies are even worse. So this is really showing that it's quite a complicated situation. And essentially shows that delta is almost orthogonal in terms of antigenic space to beta and gamma. And this makes sense if you think about that there are very different mutations on the receptor binding domain. So I won't go through the details of this, but this is unpublished data. We've done a large number of structures, probably about 15 or something, for a set of antibodies now that have been isolated from people infected with beta, so the South African virus. And in general, the number of potent neutralizers is very similar to the pre-pandemic response to the pre-pandemic virus, but the focusing of the responses again in response to beta infection is marked. There is a strong focusing, especially on antibodies that bind in the 501 region and the 484. So the responses are particularly focused on the regions of the mutations. We don't know the reason for that. The 353 public gene is almost gone, but not completely. And the 353 that is found is really broadly cross protective, very, very useful. But there's a new public gene family that's come up, which has never been picked up before, 439. And there's lots of representatives of this, and that focuses on the 501 region. That should be so there, it's quite a different sort of response in good response to viruses that have got just 3 mutations in the receptor binding domain. The good news is that there are a number of really potent cross-neutralizing antibodies. So just to then try and explain this, we've tried to use the comparative serology data to produce a sort of antigenic space map, and this is Helen Ginn's work. It's actually very similar to what people have done for influenza virus. And you can see that 351 is actually, which is the South African virus, is actually on the other side of the origin from the Delta variant. And as I said, the alpha variant and the ancestral virus are sort of more in the middle, and that explains the better sort of crosstalk between those viruses. So I think what we're trying to do now is to see if we can describe these antigenic differences, the distance in antigenic space in terms of the contributions of individual amino acids within the receptor binding domain, and that's ongoing work that Helen's doing. And I hope that would then allow one to ultimately predict how serious any given mutation would be. And I'll stop at that point. There's a large number of people being involved in this work within my group. Ren, Darming, Liz, Helen, and earlier in the project, particularly Yu Guang and Guido, have been really heavily involved. Helen Ginn from Diamond Light Source, where she's a postdoc, and the teams at the beamlines and microscopes. There was mouse work done, which just shows that those potent antibodies that we found are effective at including prophylactically at protecting in a mouse model. And Gavin's group has been heroic in this, particularly Nui, Ju, Ray, Chang, Caesar, and Ake. So it's a really, really tremendous effort from them. And we've also had collaboration with Sarah Gilmert and Tess Lamb. Other people within the NDM have contributed. And I'd just like to single out Richard Cornell, the head of the NDM, who's really helped bring people together during the pandemic and really engender an excellent attitude of collaboration. And Andy Pollard of the Oxford Vaccine Group. We've been funding from the MRC, but also from the Chinese Academy of Medical Sciences and some sort of donations that have done have helped with this. I think the open question at the end is really to what extent have we seen the worst the virus can do in terms of escape? And I think that we don't know. But let me just say a little bit about that very quickly. What I've done here is to, with input from Helen, is to map the ACE2 contacts to the RBD in a slightly different way. And that's now is looking at the antibody contacts. And again, there's a strong sort of correlation with blocking ACE2. But what Helen has done is to look at the mutational data from COG UK, where there are now hundreds of thousands of sequences. So if you say that a mutation occurs more than 10 times independently, then it suggests to me that that mutation can produce a viable virus. This is what you find. And if you put the frequency, the number of mutations, number of times a mutation is observed in the database at 10, you see you have a very large number of possible mutations, particularly enriched in the ACE2 binding region around here. Obviously, the variants really stick out like a sore thumb, as you'd expect. But the net result of this is that if you merge the contact data with the mutant data and say that the number of times an antibody contacts a residue is likely to be a measure of the antigenic importance of that residue, you can then, by putting these two data sets together, you can see those residues that can mutate and those residues that can have a potentially a large effect on the antigenicity. And that I won't go through the details of this, but there are still, this is a filtered list now of residues where that method would suggest that they might be something to worry about. And you'll see within this list, all 5 of the existing variants come up. But the thing that's interesting is that there is a large number of other variants that have not resulted in viable variants of concern to date, which may still be worth worrying about in the future. So there's a lot of caveats to that, but this this modeling, I think, makes pretty good sense. You can see here that the mutations are, if they reduce the number of possible contacts with the antibody, essentially they're rendering the RBD much less visible to the immune system. So that's, I think, where we want to try and go. We want to see if we can use the structure to be a bit more predictive and rather than just being reactive and explaining things that have already happened. And I'll stop at that point. Thank you very much.
Thank you, Dave. That was absolutely stunning, stunning journey into the interactions that drive SARS-2 binding, neutralization, and also how structural biology has been a key component of the scientific effort. As you know, I mean, getting testing going and zero surveys, etc., have their value, but it's really basic science that really drives forward the field and looks at solutions that are possible, and also provide solutions which are really important in this case because the virus is evolving so fast. Dave, if I can have your permission, there are several very interesting questions. I will go through them one by one if you wish.
Yeah.
I think that would be very nice because the audience here is very heterogeneous from parasitology background as well as structural biology and virology background. So I'll start with the first question, which in fact I also wanted to ask you, but I will read here verbatim. From the global data, can we begin to predict if the mutational space in spike is getting saturated, or is there still room for the virus to mutate?
I think that's the next question that we want to address. So, and I think that's really why in those few rush slides at the end, I indicated how we were trying to sort of look for residues that might there might be scope for mutation. And I think what we've looked at that and chosen one or two, and we're now making pseudovirus. Of course, it's what we're not have no intention of doing is to make gain of function viruses in this case. That
Yes.
in my wouldn't be ethical. But we do have a pseudovirus, which of course is completely safe, where we can do a sort of surrogate of that. So we will test that. But it is a difficult question. One of the interesting things is that there is a work from Gideon Schreiber's group in the Weizmann, where he shows that mutation at a residue, 3 residues away from residue 501, appears to increase the affinity for ACE2 to the sort of picomolar level. It makes it a very strong binder. So if this is true, and it looks as if that mutation is only tolerated in the presence of other mutations, if that is true, and what we've done that pseudovirus experiment, and the virus is not completely dead, so it does, it is viable. If that's true, then to my mind, it means that the virus would then have the ability to introduce less, some deleterious mutations elsewhere and compensate and still survive because it's got a very strong anchor on the ACE2. So I think it's a difficult question to answer.
Right. Yes. It's yes. We see maybe perhaps too early to address this issue. It seems that there was an intermediate lineage from Mexico, which came up in April 2020, which is somewhat similar to Delta in terms of the RBD. That hasn't been mentioned. I mean, that's okay. That's not been mentioned. But do you think that the Mexican lineage and the presently the Delta lineage, for example, they have independently arisen, or it could be that they're actually being sort of transported from one place to the other?
I really don't know. And I mean, there's clearly quite a large number of independent lineages around. But yeah, I mean, I guess one thing I could say as an add-on to the previous question, actually, one of the things I didn't talk about is recombination. So one of the things we should be worried about is that coronaviruses do recombine a lot. So that would be a way of bringing together, bad bits of different variants if there were co-infections. So I think we shouldn't be complacent. If I think back to September last year, people say, "Oh, there's not going to be any variants that we need to worry about. It's all fine," and then the world changed. So, I think we have to be very careful that we don't make that error.
Right. Yes. Just a few more. So we've been looking at the neutralizing binding, neutralizing antibodies against the binding regions. But there's one question: is it possible that some of these enhance infection? Because apparently there's a report in Cell or Cell Press of the same. So and neutralizing body enhancement, binding enhancement of infection.
Yeah, that's something that we were worried about in the early days, and we looked very hard to see if there was any evidence for enhancement of the antibodies we had, and we could find no evidence. So since then, we haven't looked hard.
Right.
I'm not aware of it really being a major problem.
Right. Just a few more questions, Dave. There's a very specific question. How critical is the glycosylation of the RBD at say 501 or other sites for ACE2 binding and for infection and transmission? And will it have an effect or impact on the N501Y mutant? Because that presumably will not be, well, a different pathway for glycosylation between asparagine and tyrosine. Yeah.
Generally, mostly there's not a huge amount of glycosylation of the RBD. There's a if you look at the changes in mutations that introduce glycosylation changes, most of those are in the N-terminal domain. They're quite commonly found. And I think they hide antigenic sites there. Ages ago we thought we'd found a minor variant in some early sequencing that changed changed the sequon for the glycosylation in the receptor binding domain, but that didn't hold up. I think it was a sequencing error. The sugar I don't think it's directly sort of important for the ACE2 binding. It is important for the antibody responses. So one of the therapeutic antibodies, S309, the majority of the interactions it makes are with the sugar. And we found the same with a very potent antibody raised that is elicited by the beta variant. We found one that really clamps on to the sugar, but they're in a minority. So I think really it's an open question whether there is scope for, again, more ringing the changes on glycosylation of the RBD as an escape mechanism. We don't know.
Right. Thanks. I think I'll ask the last question on my behalf. There are several other questions, and I will encourage people to send them to me, and I will transmit them to Professor Stewart for an answer later on by email. Some of the questions that have been asked have actually been addressed in the talk, and so perhaps one can discuss with one's colleagues. Just two quick questions, Dave. The first one is there a case for switching vaccine to the Delta variant? Should that be the base for a vaccine? That's question number one, very briefly. And the second is, I think you answered it, but nonetheless, we see mutations in the RBD, but alongside there are mutations in other regions of the spike. Why is that so?
I don't understand what the mutations elsewhere are doing. So some of the NTD mutations will eliminate some neutralizing antibodies. There is a feeling, but the data are not as solid as well. I don't think the data are hugely solid, but it's thought that some of the mutations elsewhere actually have an action at a distance effect on altering the presentation of the receptor binding domain. As I said, the for many of these antibodies to bind and for ACE2 to bind, the RBD has to be in the up conformation. So altering the dynamics of that will affect the antibody responses. So people think that some of the effects are sort of allosteric effects elsewhere that have a big impact. And then the other question is very interesting about vaccination with Delta, and I didn't have time to present it, but we've got some unpublished data that helps address that question. You know, because as I said towards the end, if your Delta responses alone would give you very poor protection against the South African and the Manaus strain, beta and gamma. That's inferred from the data I showed, but we've got some unpublished data that demonstrate that directly. So that would be potentially dangerous. However, we've also got evidence that shows if you're infected with Delta and then vaccinated with an early strain, then you get, or the other way around, you're vaccinated and then get infected with Delta. The net effect, there's no antigenic sin visible. The net effect is to really strengthen the responses across the board against all variants. So I think the idea of boosting, for instance, with Delta, you know, might be a very sensible thing to do.
Right. Yes, that would be, yeah, I mean, that's what I was considering, whether, I mean, to cover the variants so far, whether that can be in the pipeline for future. Well, Dave, thanks ever so much. It's been more than an hour since you've been with us, and I should tell you that is by far the most successfully attended seminar. We had close to 350 attendees from across India. And many of them have questions that have not been answered, but I'll try to convey them to you by email. We've started this distinguished lecture series, and you are the first speaker for it, and you've really set a very, very high benchmark and for our institute to try to maintain speakers at this level of scientific excellence. But thank you so much for that, for your time, and it was a pleasure as always. And if and when you happen to visit India, you have to visit this institute. Thank you very much.
Thank you. That would be lovely. And I will, if you send the questions through, I will try and answer them. Yeah.
Thank you.
If I can. Okay. Thank you very much.
Thank you. Bye-bye.